Okay, I think we are good. To everyone online and here, thanks for having me. My name's Kevin. I work in the Genomics Research Technology Hub over in Sprague Hall. I started in August. Been here a couple of times for these meetings, but excited to talk today about building reproducible single-cell workflows with Snakemake, and hopefully it's applicable for anyone who wants to use Snakemake or workflow management systems in general. Um, So I'll talk about the problem with pipelines in general and especially exacerbated in single cell workflows, and then some of the benefits of using a workflow management system like Snakemake, chiefly automation, ensuring reproducibility, scaling efficiently, and reporting results. So the problem with pipelines in general and single cell pipelines specifically is that they are complex. They involve multiple tools, languages, and analysis steps. They can be difficult to reproduce, and they can be computationally demanding with HPC resources or GPUs and certain steps requiring vastly different resources. And they can be error prone, especially if they involve manual execution. Uh, they can also be hard to scale. That's coupled with that manual execution where adding new samples means repeating everything manually. Um, and that's adding new samples is very common in any sort of, you know, bioinformatic analysis. So the solutions that Snakemake brings to those problems is that allows you to automate. So for single cell data, going from raw sequencing to clusters to visualizations, key point here is with clear declarative rules. I'll come back to that. Um, you can ensure reproducibility by locking in software versions, maybe with Conda or with Singularity containers on HPC or Docker or whatever you might be using. You can scale efficiently by running tasks in parallel, and the key point, by skipping unchanged steps. And you can report results as part of an integrated pipeline. So there's no sort of discontinuity between the work that's done and the work that you report you did. um, so that first benefit of automation, that automation is achieved by clear declarative rules. What does declarative mean here? So I'll contrast declarative with a traditional approach toward running a pipeline. Maybe you're running a simple pipeline in a single R or Python script, and you specify each step to be run. One after another. And when you need to rerun any part of that pipeline, you need to rerun the entire thing or manually keep track of different versions of things. So that would be then an imperative approach. And in contrast, the sort of a declarative approach here is that you specify what you want at the end, and you use rules, and in Snakemake, literally use that uh, protected word rule, and you Uh, so the rule I'm showing here is a special rule which specifies your final outputs, but you can imagine further down in this file are individual rules for each of your intermediate files. And then you let Snakemake determine the sequence of rules that need to be executed uh, as so it determines its execution plan. I understand that this could be kind of vague at the moment, but hopefully I'll get further into it and it'll be very clear. So one more time to reinforce that. Typically, you would think in a script, you would take a top-down approach where you would run step one, step two, step three, step four, all in sequence. And if step three changes, you rerun the whole thing, running one, two, three, four. Whereas if you use Snakemake or another similar workflow management system, you're going to let the workflow system itself figure out the relationship between all the files in the workflow so that if any one of these files needs to change, only the steps that need to be rerun get rerun. And it does this, Snakemake does this, and every other system does this. Nextflow is a similar alternative in bioinformatics. There are many others outside of that field by building a dependency graph to determine which rules depend on which other rules. So here's an example of the dependency graph for the pipeline that I'm going to be describing here today. This is a parse single cell analysis pipeline. This dependency graph is also sometimes called, or it's in the form of a directed acyclic graph or a DAG. You'll sometimes see that term around, and it's basically a flow chart. If you're totally unfamiliar with either of those terms, which just defines the relationship between the rules, like I say. And I say directed because you can see the topmost rule, that rule all, excuse me, we're looking at backwards. The bottommost rule all depends on each of these prior rules here in a single direction. There's nothing looping here. Okay, so I was torn in the course of this talk whether I'm going to talk about all the benefits this workflow system can bring or teaching Snakemake. I'm not going to be able to teach as much Snakemake details as I would like to here, but I'll show little snippets as I go. This little snippet here shows the final outputs that I want from this single cell analysis pipeline. So I've got a final report. I've got a ScanPy H5AD file combined for all of my samples. And I've got individual outputs from running the parse processing software that's called Split Pipe. It's similar to Cell Ranger if you haven't worked with that data set before. So this is the top of my Snakemake file called a snake file. And when I want to run this snake file, I go to the terminal and I execute Snakemake, and then I specify the number of cores on my machine, and then it runs through the whole workflow. It discovers all the samples present, it runs the steps in the correct order, and it executes independent samples in parallel, and it skips completed steps. If I needed to add a new sample, I would just add that new sample into the input directory that I'm using here. This slide illustrates, so automatic sample discover is a feature of these workflow management systems in general, but here those samples, I'm finding those new samples by means of a Python function. And this illustrates my main bias toward using Snakemake compared to other workflow management systems is that it is just Python. So you can use any functions, you can import any Python modules that you want. So in my snake file here, I had this function to discover any new samples present in my input directory. That's the FASTQ directory here, and to assign them into this wildcard called sublibraries. So I'll get into the meaning of wildcards now because that's a key point of how Snakemake is able to intelligently discover which steps need to be run. I sort of described it as a bottom-up approach before, where you have your all initially, and the key, or one key part of Snakemake, is the existence of these wildcards, which are discovered sort of backwards from your output files. So you can sort of see, now I'm going to jump back a little bit. I didn't get, you can see here in my final output, what I wanted was, I used the special, this function expand to look for directories with this wildcard present, this sublibrary wildcard. And then I specify here to be the sublibrary key to be sort of plugged in for all of the sublibrary values in my workflow. So Snakemake is kind of working backwards to discover what the values of the sublibrary wildcard should be, and then executing all the rules to construct this intact DAG. Right. Uh, Another part of being declarative and being explicit about the contents of these rules is making use of this configuration files. So in Snakemake, that is accomplished by means of this config.yaml file, which is a common feature of any Snakemake workflow you might look at. And here I finally get a chance to kind of dig into the structure of a Snakemake rule. So I think this step might be familiar to many of the people here. This is a step to integrate all of my individual single-cell RNA-seq data sets together. So I have rule, and I'm with this meaningful name. I'm specifying my input files, and those input files are all the results of my parse sequencing analysis with that split pipe software. So the input to the integration is the output of a different rule. And then the output of this are individual, a single ScanPy H5AD file. I'm specifying a conda environment. So that ensures that, I can keep track of which dependencies, which, Python modules I might be using, or packages I might be using. And then I have individual parameters that I'm plugging into the code that is actually executed here. And those parameters are all derived from a config.yaml file here, where I'm running config.get, and this is just a dictionary with keys. So one key is the minimum genes I want to use for this processing. The threshold by which I want to subset my data, the minimum cells, the number of top genes I want to use for finding highly variable genes, etc. Going one step down, this is a bit of a preview, but we have a section for the number of threads I want to use. So I'm specifying my computational resources, and then the memory and the number of CPUs I want to use. I'm doing all of this on HPC here, and then the actual command. So a bit of that was an interlude, but I'll keep moving on. So that first step, the second step here to ensure reproducibility, the key means is by being able to specify our environment explicitly within these snake files. So common problems here is that, it worked last month, now it's broken when software is updated, or it works on one environment or not another, or software is not sufficiently documented because you're relying on sort of manual documentation, or you're missing dependencies. So solutions for all those problems are present if you're using a workflow management system. You can have isolated rules with different software versions. You can document the specifications within your code in the workflow, and that all of that ensures can help ensure reproducibility, especially by means of making use of conda or containers. So you can declare within individual rules that rule use a particular environment. That could be a conda environment, that could be a singularity container or a Docker container or whatever. And you can specify that an entire environment, the complete details of a particular environment within your workflow, so that is built in the course of the workflow if you really wanted to do that. And all of that can be enabled at the command line by just asking Snakemake to use Conda or Singularity or whatever if you cared to do that. But similarly, you can use pre-built Singularity or Docker containers. And importantly, for anyone who's using HPC, I'd love to help people do that if they're set up to work on that system. You can load modules within individual rules. Getting further into like specifying dependencies, you can, as I detailed earlier, specify details of individual rules within a single configuration file. And in my case, I'm just showing for demonstration. What I usually do is keep one configuration file for a given pipeline, and then have a project-specific configuration file so that I can manage slight variations when I'm running the same pipeline on different data sets or in a different context. And then all of that is can just be loaded automatically within the snake file you might be working with. A key part of my workflow is that all these pipelines are version controlled with Git. That's straightforward to do. So that's also kind of documenting not only your code, but also the infrastructure to some extent. You're specifying all your dependencies if you're able to set that up. The third benefit that I see from workflow management systems and Snakemake specifically is being able to scale efficiently. And that's basically being able to move between different systems and being able to run tasks in parallel and skip unnecessary steps. So when Snakemake goes and builds its dependency graph and discovers that there are multiple, maybe you have multiple samples that you all want to process in identical ways all at the same time, those different rules can all be run in parallel. And then steps that need to be run all together, for example, in this parse single cell sequencing situation, it's necessary to individually process particular sublibraries of your data and then process all together once you've generated all of those outputs. So it's fairly simple to construct that kind of logic within the workflow that would be much more challenging to keep track of manually. So there's parallel execution and intelligent re-execution, where if you have run a workflow to completion and you try to run it again with Snakemake at the command line, you'll get a response that there's nothing to be done. All the requested files are up to date. That could be means of either modification times of the files that you're specifying, or by the actual hashing of the outputs. You can adjust that within Snakemake. If in fact you added a new sample and parts of the pipeline needed to be run again, Snakemake would only process the new sample, and it would rerun any steps that would combine multiple samples. So in this pipeline, we're rerunning the parse combine, we're rerunning integration. And then if I changed the integration parameters for this single cell data, then I would only run that step. So yeah, I just mentioned that these dependencies can be tracked by means of file modification times, or by changes in the code, or by changes in the parameters that you're setting in your Snakemake workflow. Right, so here's an example. If I changed in my parameter setting from 200 minimum detected genes in my integration step to 300, then only that step would be rerun. And it would be rerun every time I change that value. So I haven't investigated thoroughly the capabilities of, other workflow management systems in this regard, but to me, this is a major advantage for Snakemake, as I'm working on HPC all the time, that Snakemake makes it fairly straightforward to integrate with Slurm on HPC, the Slurm job scheduler. And all that's really required to set up a Snakemake workflow to work in Slurm is to specify within individual rules various different parameters for Slurm submission. So you would specify the partition, the account, maybe the runtime, and the memory and the CPUs. And then you can execute Snakemake specifying at the command line saying executor Slurm, and Snakemake will submit each individual rule as a separate Slurm job. So that's, that seems like a major advantage to me. You can, be very explicit about exactly what resources are required, and then it would vary greatly in this pipeline. For example, the integration step is going to require a huge amount of memory, so it's useful to be able to specify that here and to ensure that that's submitted separately from the individual processing steps. And then, yeah. So that Slurm, you can run Snakemake via Slurm at the command line, as I said, by specifying executor Slurm, but you can also specify a sort of user configuration file, and the location of that on HPC is going to be this .config Snakemake, the name of your individual arbitrary name for your configuration, and then config.yaml. And then you can specify various default settings for any Slurm submission. Then it's very straightforward to always use your own lab's account for your job submissions by specifying the profile at the command line when you run Snakemake. So here I'm running Snakemake profile Slurm and using Conda, and I can have the whole pipeline run while I go home. Well, that'll only happen at the end of the day, but and yeah, I already got into it in this situation. For any individual rules, you can specify the resources you need, which is going to be critical for Slurm submission. And you could even specify, and I have done this in some cases, using CPU or GPU partitions. For example, if you happen to be able to speed up some small portion of your pipeline by using the GPU partitions, which I think are otherwise often underused, you can do that and not have to write separate steps. In my case, and this is fairly straightforward to do, I also set up monitoring of progress via email. This is just for start and stop of the job, so I can tell if something fell apart and I can restart it if it's going to be several days that it's going to be running. And as with any other Slurm job, you can monitor the progress of jobs with the conventional Slurm commands. The last benefit I will lay out here is the ability to report results. So I am reporting results in Snakemake by defining a separate rule to generate a report using Quarto. We don't necessarily need to get into all the details of how that works. If people are unfamiliar with how Quarto works, it's basically a way to make HTML files that describe, I made these slides with Quarto, for example, to specify them with code. So the advantage of this integration of reports with the workflow is that there isn't a disconnect between what you say you did and what you actually did, because you're eliminating that sort of manual step. So here I have, as with all the other rules that I described, certain input files that I expect to be generated before the report is made, an output file that is just this HTML file, and certain parameters that I'm using here. So this is like a template document that I would provide additional parameters for, and then certain code to render this report. And I would get and here's that template I'm describing. And in the output, I would get a final HTML file that I could send to someone that say, hey, this is what I did. Additionally, you could have that custom report that you would generate describing what you say you did. You can also generate automatic sort of runtime reports within Snakemake that describes exactly which rules were run, how they relate to each other, and how long each step took, so that you can easily sort of debug problematic, uh, problematic steps in your pipeline. And you can track resource usage and, file provenance and ensure that all the parameters that you thought you used were actually the ones that you used in in your pipeline. And you could even go so far as to generate, build little apps or dashboards if you care to do that. That's maybe a bit much for most most projects, but it's possible. So to put it all together, in this example single cell workflow, we have a snake file that defines all of our rules, a config.yaml file that defines various parameters that would be used in the workflow. And then this is not a hard and fast setup, but in my case, I would have a data folder that would have all that input data where if I added a new sample to this folder and then rerun the workflow, that new sample would automatically be run. I could specify environments that describe the conda environments that I'm using and any arbitrary code I might use within any individual Snakemake rule. I could specify reports or the location of reports to be output, and then a output directory with intermediate files. And all of that output would be generated through this Snake file that I have here. So with arbitrary Python code and then individual rules for foremost that rule all that specifies the final outputs that I want to get, and then individual rules that I'm not going through here that describe each of the inputs and outputs of each step. Running this workflow is as simple as navigating to that directory that has the snake file and running Snakemake. You can run Snakemake with a dry run to confirm the dependency graph that you're going to use. And then you can specify a certain number of cores to use when you actually want to run the main event. You can run it straightforwardly on Slurm by specifying either a configuration profile Slurm or running it with this executor Slurm and providing all these further Slurm configuration details. And you can generate a final report to describe what you did. So since we've made it through all that, I have a few extra points that I think make workflow management systems in Snakemake especially useful. And that is that when I structure code in this way, I think that it is very tractable for modification with LLMs, with code assistants, because it's very straightforward to specify the context of what you might be doing. It's easy to say, like, work on this rule. Why doesn't this input or output file exist? And it's very easy for these tools to parse these sorts of highly structured pipelines, as opposed to navigating a sort of complex, unfamiliar code base organized in all sorts of different ways. And it's also very easy to evaluate whether certain modification has sort of broken the pipeline, because it's easy to inspect that dependency graph and see, are the changes I'm making going to break the pipeline without actually having to run any time or memory intensive code. And finally, another reason why Snakemake in particular is good for use with AI and LLMs, and in this, I'm talking about like Claude Code or that sort of thing, is that it's written in Python, and the code base, or this is just extremely familiar with Python compared to many other languages. And then, so specifically why I think that these workflow management systems are useful for editing via these AI or LLM tools is because it's very straightforward to constrain the problem space, to provide context, and to evaluate the validity of any changes made. Feel like I've been really rolling here. Anybody have any questions? If not, I'll wrap up with some of this stuff and I can get into some like actual, yeah.
I have a question. So if we were to change a script that's inside of our pipeline, will the pipeline run automatically will pipeline run that script again, even if we haven't changed any of the outputs yet?
Yeah, it depends which way. Yes, by default, it'll work. It will observe, Snakemake will observe changes in the script. You're saying the script is part of a rule, like
Yes.
yeah. If you change the script, yeah, by default, it would rerun because it would detect changes in the file modification of that script. But you could, if the changes you were making to the script, you were sure were not going to change your output, you could uh, yeah, you could touch the output, like the command in Snakemake is snakemake touch file name, and then you could prevent Snakemake from reevaluating the output. Yeah. Um, so yeah, to sort of re lay it out in a sort of quick start guide here. If you wanted to work with Snakemake on your own laptop, uh, or even I suppose on HPC, but let's talk about it. A laptop here, and you had installed Conda. That's the recommended way they say to install it. You can pip install Snakemake, but probably sensible to make Conda environment and then and install Snakemake here, and then activate that environment, and you could make a directory and make a snake file and a config file and some particular project format, whatever one you might use. Edit that config file, put your data in your data directory or in a FASTQ subdirectory, run your pipeline in a dry run mode to evaluate what steps are going to be run, and then run it in an active mode, specifying your cores or specifying profile slurm. And there are just tons of available pre-made pipelines that you could modify yourself. I'll show some resources at the end of this. And there's if you go to GitHub and search for Snakemake or Snakefile, whatever domain you might be working in, there's lots of examples that you could probably quickly modify to your purposes if looking at a blank Snakefile is scary. To summarize, Snakemake useful to automate everything, to ensure reproducibility, to scale efficiently, and to report results. Those resources, for one, is the Snakemake workflow catalog. They have pre-made workflows. I've struggled a bit to get them to work because they kind of automate so much that I can't tell what I'm doing. But there are lots of GitHub repositories that could be quickly modified. There's documentation for Snakemake. There's the Carpentries has a good lesson on using Snakemake, and there's the usual places you could Google. But the docs for Snakemake are really pretty good. If you wanted to use another workflow management system, as I said, Nextflow is an alternative. They have a lot of pre-made workflows as well. It works on a slightly different model that I'm not going to get really into here, but it's similar in concept. This particular workflow is one I've used. This is built for bulk RNA-seq, but this is something that is fairly easy to read. It was published in 2019. This ARMOR workflow is a Snakemake workflow. It's pretty easy to modify. And you can make your own, share them on GitHub. And Snakemake's useful for small projects if you just want to keep track of documentation and remember exactly what you did. For medium projects, to be able to scale up individual rules, or for very large projects if you need to keep track of a huge DAG and need to be sure that every sample ran the same way or in the way it was supposed to. Uh, a lot of restatements about that, but I think that does about it. Do we have any questions? More?
Yeah, so I have a question. So if you are managing multiple projects, would you recommend one snake file per Snakemake, I guess? Or like one snake file per Snakemake instance, or would you create new ones?
Yeah. My usual approach is to, if it's a given like situation, like if I'm working all with parse single cell data or all with 10x single cell data or all bulk RNA-seq, or then I make a single version of the pipeline that works with one of them and version control it. And the thing, if I can't capture all of the differences within a single config file, like just variations in the parameters, if it's like dramatic changes, then this wouldn't qualify. But I have like a local configuration file for each project so that if some massive change in the logic of the pipeline happens, if I discover that there's some new version of cell ranger that demands the input data be a certain different way, then I can just change the pipeline and commit it and be sure that it's the most up to date. And any projects I've run in the past, that version that's still present in that repository is accurate to what I did then, but sort of have a template that's version controlled and then have sort of different versions of that if it's an individual project. But if for vastly different modalities, I would have different snake files and different setups. Yeah. Yeah. Yeah, I was really rolling. As I say, I toyed between getting really into the nuts and bolts of how this works with examples, and we could do that if people want to get into it. I don't know how many questions people would have. So I just wanted to show like the benefits from my perspective. Uh, Um, but I haven't been checking would it be Slack or Zoom if anyone actually, no questions there. Okay. Yeah.
Wanted it, I'm saying, for someone that is just trying to analyze some single cell data, do you think they should like take a Snakemake workflow from someone else and like they change things here there, or do they just, use their own scripts and make their own Snakemake?
Yeah, I don't know how much I'm using I use like GitHub Copilot and Claude Code often, and my suggestion would be to use an existing template, just ask it, make these changes, and then look at what it's done and figure out and like try to understand it from that perspective. That would be my first suggestion. If you're not interested in getting into those, then yeah, just take an existing repository and go through it, step by step. There are many single cell instances of Snakemake profiles, but even as I said, that armor pipeline from 2019, I think it's Charlotte Soneson. That's a great example. It's very easy to follow. It's designed for bulk RNA-seq, but it's pretty adaptable. And the essential steps of like that initial processing and running fast QC and multi QC are going to be the same. Yeah. Yeah.
Make sure I'm understanding. I feel like what I'm getting from this is that if you have a pipeline that needs to be modified for different use cases, it's easy to do that with Snakemake.
Yeah. Yeah, I would say so. Um, I think that it's a challenge not to get into I'll pull that one up just the heck of it. Where are we? So the example I actually built up that I started with and decided, no, I should really actually talk about single cell data. not and not get off track, but since we have a little bit extra time. So the example I started with was like a text analysis of Romeo and Juliet, which I just adapted from an existing Snakemake tutorial. I don't want to steal any thunder, because it's fairly easy to keep track of like what's going on. We can state in simple words the analysis that we're doing instead of using a lot of terms that maybe people aren't familiar with. So I just wanted to get across that like the structure of having that rule all and having individual rules that build that graph is pretty easy to appreciate in this example where we were counting the words and lines in Romeo and Juliet. We're counting the occurrences of Romeo or Juliet. We're counting the 10 most frequent words in the text and making a little report. We can write individual rules. And if I sort of step through this, you can see that I have you can see that dependency graph being built up. So in this example, our first step would be to download the data. So go pull it from some URL. You can see here this rule download data. We have an output that is a text file and a parameter that's a URL, and then a command that is just using CRL or wget. It could be wget, but CRL here. And we have a graph that is one part, which says download data. And then we could do another rule to count the words. And then you can see that graph extending by one. Now we have first download data, then count the words. And then we could have a, right. And then so we could have wildcards that would allow you to operate on multiple texts. So in this example, first I downloaded Romeo and Juliet, but then maybe I also download Midsummer Night's Dream, and I download the data and I count the words in Romeo and Juliet, and I also count the words in Midsummer Night's Dream. And then, oop, there's a lot of, what happened to my tags? No, I lost all my tags. I'll just show it here. Oh, yeah, here. Ooh. So we might, build up this graph step by step as we add additional rules. So we have our rule, we're counting all the words, but we want to count all the matches of Romeo and Juliet by counting each word in each line. And then maybe we want to count, we want to count actually the most frequent words. And we also want to avoid the words that are going to be like and, I, or but, or whatever. So we build another rule for that. We can compile a report, and all, this complex logic has been encoded in a simple, readable snake file that we can jump to any individual part and modify those changes. So that's a long answer to, I guess that was your question, to how you can easily modify, and it's very flexible.
That helps a lot. Thank you.
Oh, great. Oh, cool. I don't want to take everybody's time. It was over a little quickly, sorry. But there's a lot to get into. So yeah.
Next question.
Yep.